The environment we live and work in is not the environment our senses are made for. And this mismatch between the modern man-made environment and the natural environments that shaped our senses can have some harmful consequences. If we look at the evolutionary history of humans and apes during which our visual system was shaped, the Industrial Revolution, which brought all the buildings, urban landscapes, and modern artifacts that make our visual diet today, is an extremely recent event. Human history, since the Industrial Revolution, represents an insignificant stage in terms of sensory input. To visualize this, let's represent the modern environment proportionally to its duration during the last million years. Insignificant. What we will see in the next minutes is that understanding the differences between the visual properties of natural environment and this tiny part of our visual diet, tiny if we consider evolutionary times, so understanding these differences does help us understand better visual discomfort. So in particular, we will answer two main questions. What are the visual features that make a picture uncomfortable to look at? And why are these features detrimental? Natural scenes, from tropical forests to Mediterranean woodlands, from deserts to savannas, have a considerable diversity. However, they share some regularities. Luminance, or intensity, tends to vary smoothly. Neighboring locations are very likely to have similar luminance level. This means that low contrast between neighboring locations is much more frequent than high contrast. But we can go further than that and show that the correlation between the luminance of two locations varies consistently with the distance between these two locations. To understand what this means, let's look at the profile of the variation in luminance with space in a sample. So this profile corresponds to the top of the curve below. Here are the profile of the luminance. And the top curve can be made of a sum of various curves with different frequencies, spatial frequencies, amplitudes, and phases. This is the basis of Fourier analysis. If we add here the first, second, third, fourth, and fifth curve, we get the top curve, which gives the luminance of the sample. So here, the first curve corresponds to low spatial frequencies, and the fifth curve to high spatial frequencies. So a lot of wiggling variations in luminance. If we plot the amplitude of curve 1 to 5 in logarithmic coordinates, we see a decreasing straight line. This graph is called the amplitude spectrum of the image, and this linear decrease in logarithmic coordinates is called 1 over f. What is important here is that this 1 over f structure is very consistent across natural scenes, tropical forest, savanna, dry desert, in spite of their apparent diversity. Why is that important? It is important because, according to the efficient coding hypothesis, sensory systems have adapted to deliver a representation of the natural environment that is efficient. Efficient both in terms of information content and in terms of metabolism. The efficient coding hypothesis has received strong empirical support at the level of properties of single neurons and at the level of a whole system. In human perception, for example, we know that discrimination and detection performance are enhanced for stimuli that have a 1 over f structure. For visual discomfort, a lot of studies have shown that deviation with respect to the 1 over f statistics create discomfort. So for example, if we take noise with a typical 1 over f structure and you change the structure to have a steeper slope, so an increased representation of low spatial frequencies, or a shallower slope you get more visual discomfort. But we can improve on that. So we can improve on that by considering the two-dimensional structures of images. In the same way as you can decompose a one-dimensional signal into a sum of simple sinusoidal signals of different spatial frequencies, with Fourier analysis, you can do the same for signals that have undulation in 2 dimensions. And if you do that, instead of having a single slope, what you find is that to get a cone, a regular cone, in the 2-dimensional Fourier space. So this gives a family of 2-dimensional cones representing the typical fall-off of luminance correlation with distance in natural scenes. To measure deviation with respect to this reference, you first compute the amplitude spectrum of a scene, and then find the best fitting cone in the family. You next look at the magnitude of the difference, which gives you residual numbers. When you sum these residual numbers, you get a single measure of deviation from natural scene, with no fitted parameter. So this image of a working environment gives a high number. Why? Because the building is made of regular modular units that are associated with a strong contrast at some spatial frequencies. These frequencies dominate in the Fourier spectrum and disrupt the 1/s spectrum typical of natural scenes. It turns out that this single number that measure deviation with respect to natural scenes predict a good part of the variance in judgment for discomfort, therefore providing a good predictor of the discomfort a scene can generate that can be computed very easily. However, such a characterization of adverse images using the statistic of natural images and Fourier analysis does not tell anything of the possible mechanism underlying visual discomfort. For uncomfortable images, a question therefore naturally arises. What is the specificity of the underlying neural activity that makes them uncomfortable to look at? There is growing evidence from neuroimaging studies that uncomfortable images produce an excessive response of the visual cortex. On the computational level, Haigh and Olivier have shown that sine gratings, which are visually uncomfortable to look at, produce a larger and less sparse response in a bank of Gabor filters. The next question concerns the differences in susceptibility to visual discomfort between individuals, differences taken to an extreme in clinical population. What properties of the early visual cortex could explain that an apparently safe image is aversive for a hypersensitive individual? So, there is some evidence that such differences may be driven by a different balance between excitation and inhibition, with potentially reduced GABAergic inhibition leading to hyperexcitability in the cortex. So we address these questions on the activity underlying visual discomfort using a mechanistic model of the visual cortex. The model used is a firing rate model based on a recurrent excitatory inhibitory network with a columnar retinotopic organization. This model has realistic classical and extraclassical receptive fields and a realistic pattern of lateral connections. The model accounts for several fundamental processes emerging early on in the visual pathway, such as control enhancement, ISO orientation suppression, and figure ground segmentation. So using this model, we saw that uncomfortable images were associated with a dense activity of the model. That is to say, a larger number of units active and with a larger activity or firing. So this is represented here by the red curve. And in contrast, comfortable images were associated with a sparser activity, with only a few units firing, and this is represented here using the green curve. So we found that the sparseness of the model response to an image predicted a large amount of the variance in judgment of discomfort. We also studied how the activity of the model changed when the ratio of excitation over inhibition was modified, and when this ratio was increased. So therefore, modeling a limited availability of GABA-depending inhibitory activity. We saw that with less and less inhibition, there was a systematic drift towards a denser activity of the model, in agreement with an increasing load of the visual cortex. So in summary, we have seen that a neurodynamical model of the cortex predicts psychophysical judgment of discomfort, and comfortable images are associated with a denser, less sparse activity, in agreement with a putative link between visual discomfort and efficient coding. And the activity of the model is less sparse when the ratio of excitation over inhibition is increased, in agreement with a link between increased susceptibility and lack of GABAergic inhibition. As an interim summary, we have seen that knowing the statistical regularities of luminance in natural scenes provides a reference of what the visual system processes best. We have seen that deviation with respect to this statistic causes visual discomfort, and that these deviations can be easily computed. More elaborated models capture what could be the mechanism underlying visual discomfort and what can be the origin of the differences in susceptibility to discomfort between individuals. All what we have seen so far is about luminance. What about color? Is there any contribution of color to visual discomfort? We know that this is the case in simple geometrical stimuli. Haigh and Wilkins measure both metabolism and discomfort from colored gratings made of two colors with different separation in chromaticity, computed in the CLUV perceptual color space. Over a large gamut of chromaticities, increased chromaticity differences were consistently associated with increasing hemodynamic responses. Our first experiment about color can be summarized in a simple single sentence. Contrast self-reported visual discomfort from an image and the image average separation in chromaticity, which we are to define. So what we did was to define two sets of images of abstract art to minimize the semantic content of the stimuli, and for each set to ask observers to rate the images for discomfort using a Likert scale with values between 1 and 5, from no discomfort at all to very uncomfortable to look at. That's all for the psychophysics of this experiment. We next compare the ratings to separation in chromaticity. So let's see how we compute differences in chromaticity. The definition of the metric is simple. It exploits the association between separation in chromaticities, as measured in the CLUV space, and cortical responses in Haigh and Wilkins' work we have just seen. So for each pixel in an image and all the adjacent pixels, we computed the chromaticity in the CLUV space, and next computed the Euclidean distance of each neighboring pixel to the reference pixel. Finally, we average all these different distances, and we did that for all the pixels in an image. So when doing that, we get a single number for each image. We did that for all the images in the two sets of stimuli. So we found that average chromaticity difference predicted well visual discomfort and accounted for a part of variance that wasn't accounted for by models based on deviation with respect to the 1/f structures of natural scenes, so on luminance. The comparison with 1/f, however, is based on deviation with respect to the balance between contrast energy at different spatial frequencies and may miss the total luminance contrast energy in the image provided by the edges. To control for luminance contrast energy, we define stimuli that varied in average chromaticity difference, but not in luminance contrast energy. Starting with an image, we send all its pixels in the loop space and rotated the space with respect to its center while keeping saturation constant. However, the rotation were not usual rotations, but rotations with a non-constant angle across radial directions. These rotations deform the space continuously, but compress or blow up some sectors of the chromatic space, leading to different values for the color metric. We define a huge number of such pseudo-rotations, and for each starting image, we randomly selected 3 levels for the metric, a low level, a medium level, and a high level. Using the same experimental procedure as earlier, we ask observer to rate this triple for visual discomfort, using all the stimuli displayed in a random order. And what we found was that increasing values of the metric were associated with increasing visual discomfort, confirming the previous result with more control on luminance contrast energy. So, we have an association between chromaticity difference and visual discomfort. But can this relationship be framed in terms of the statistic of natural scenes? To answer this question, we computed the average chromaticity difference for 2 sets of calibrated natural images. We found that 97% of the distribution fell under a value of 0.01, a value not associated with visual discomfort in the 2 experiments. So this shows that visual discomfort increased as the chromaticity difference exceeds those typical for natural scenes. To better understand the extreme values of chromaticity differences for natural images, we computed the metric for smaller random samples of the calibrated images. We found that the samples with the highest value for the metric represented images of ripe fruits against a foliage. That is to say, stimuli thought to have played a central role in the evolution of primate trichromacy. So this study may provide a new perspective on how to understand visual discomfort. Let's consider the monstrous space of all possible stimuli, all possible images. A big subset of them is processed without any problem by the visual system. Natural images make a tiny subset within this set of images easily processed. This inclusion is in agreement with the theory of efficient coding. Now, our stimuli in these experiments straddle the border between comfortable and uncomfortable stimuli. The uncomfortable stimuli are somehow exaggerated versions of fruit against foliage. Or in the building experiment, these images of building made with modular construction, full of stripes, are exaggerated version, maybe, of strong contours. So this raises the question of whether it is generally the case that visual discomfort arises when adaptive perceptual mechanisms are overstimulated by specific classes of stimuli rarely found in nature. But this brings me to the conclusion. So, we have seen that understanding the statistics of natural scenes help us understand visual discomfort. For luminance, deviation with respect to the Fourier spectrum of natural scenes are associated with visual discomfort. For color, our principal measures of chromaticity differences with no fitted parameters accounts for substantial variance in judgment for discomfort. Chromaticity differences that cause discomfort are not found in nature, but ripe fruits against foliage have especially high values for the metric. So that proposes a new perspective on visual discomfort, in which that leads to the testable hypothesis that visual discomfort arises when adaptive mechanisms are overstimulated by specific classes of stimuli never found in nature. So I would like to thank Sarah and Arnold for all these wonderful collaborations, and I would like to thank you all for your attention. Thank you.